Hello, good morning, class. My name is Mr. Goodluck Nabike, but my friends call me Power. I'm a science graduate with specialization in applied biology. I graduated from Ebonyi State University with second class upper division. I'm as well a graduate of Woteso Academy. I've been teaching science for over 3 years, and I have experience in teaching physics, chemistry, and biology. I believe I have the skills and the experience to be an excellent teacher and also to help your students achieve their academic goals. I'm a strong advocate of hands-on learning, and I believe that students learn best when they use this process. I use varieties of teaching methods such as laboratory, lectures, practicals, project, and discussion to ensure that students learn and understand science. I'm very committed in helping your students to develop a skill they need to be successful in their careers and as well in the college. In our lectures today, we are going to talk about a gene and gene linkage and also Mendel's inheritance. You can bring your pen and paper out and write down your questions so that by the end of these lectures, you can be able to ask your questions. Our scope of learning. By the end of this, our lesson, we should be able to identify the structures where the gene is located in the cell using the microscopes. We can also be able to describe gene and its protein synthesis, and also to describe gene linkage and Mendel's inheritance. What is a cell? A cell is the smallest unit of structure and function of all the living things. They are grouped into two. We have the prokaryotic cells and the eukaryotic cells. When you talk about the prokaryotic cells, there are single-celled organisms. That is, the organisms that lack true nucleus and lack a specialized structure known as the organelles. They are mostly unicellular organisms like the bacteria, but they are capable of carrying out all the functions that are required in life to be carried out, such as metabolism, homeostasis, and reproduction. When you talk about the eukaryotic cells, you are talking about multicellular organisms like a plants, animal, fungi, and a few unicellular organisms that has a true nucleus and a specialized structure called the organelles. Cells are essentially carbohydrates, lipids, protein, and nucleic acid in a water-based environment. There are three major parts of the cell. We have the cell membrane, we have the cell cytoplasm, and the cell nucleus. But in our discussion today, we are going to focus mainly on the cell cytoplasm and also some parts of the cytoplasm, such as the endoplasmic reticulum and the ribosomes. So this is the microscopic view of a plant cell. This is what you will see when you use a microscope to have a view of a plant cell. These are the components of a cell. We have the cell cytoplasm, we have the chloroplast, we have nucleus, the nucleolus, we have endoplasmic reticulum, we have mitochondria. That is the powerhouse of the cell, where metabolism activities takes place. We have the perisomes, we have Golgi apparatus, we have amyloplast, we have ribosome, we have cell membrane, we have the cell wall and the cell vacuum. But in our today's lecture, we are going to concentrate more on the nucleus, which is the place where these genes are being stored, and also part of the cytoplasm like the ribosome and endoplasmic reticulum. I believe with conviction that you all can be able to describe a structure of a cell when you have a view of it in the microscope. What is a nucleus? A nucleus is the largest part of the cell. Nucleus are defined by double layer structure called the nuclear envelope. You can as well call it a nuclear membrane, which contains the DNA that carries the instruction for making proteins. Nucleus regulates the growth and division of a cell. We have ribosome. This is the site where protein synthesis takes place. I believe with conviction that you have all known the meaning of a nucleus. We can then proceed to genes. What is a gene? A gene is a DNA molecule containing biological information. There are about 20,000 to 23,000 genes in human body. Genes are contained in the chromosomes, which are located in the cell nucleus. A chromosome contains hundreds to thousands of a gene. As we know before, that there are 23 pairs of chromosome in human body. That is, we have the X chromosome and the Y chromosome, the two types of chromosomes. So, but when you bring two of them together, it's going to be 46 chromosomes in human body in total. There are four bases of DNA. We have the adenine, the thymine, the guanine, and cytosine. But before I explain in detail about this DNA, let's take a look at the DNA strands. This is the structure of a DNA strand. It's grouped into two. We have the phosphate group and the sugar group. In this phosphate group, now you are seeing cytosine pairing very well with the guanine with the help of a bond called hydrogen bond. In the sugar group, you can see the thymine pairing very well with the adenine with the help of this hydrogen bonding. This is a typical structure of a DNA strand. When you take a look at the left diagram, you can see a clearer view of a nucleus. This is exactly how it looks like when you view it through the use of microscope. This is the nucleus containing the genes, which has the DNA molecule that carries the biological information. We have the mitochondria, also known as the powerhouse of the cell. What it does is that all the metabolic activities, that's what it performs. We then move to protein synthesis. We all know that protein are probably the most important class of material in the body. They are not just the building blocks for muscle, connective tissues, skin, and other structures. They are needed to make enzymes. Enzymes are complex proteins that control and carry out nearly all the chemical processes and reactions within the body. Proteins are composed of long chain of amino acids that are linked together one after the other. 20 amino acids are used for protein synthesis. And some of these amino acids come from diet, that is from the food we eat, while others are produced by the enzymes in the body. These 20 amino acids that are needed for protein synthesis to take place in the body are grouped into three. We have essential amino acids, we have non-essential amino acids, and conditional amino acids. When you are talking about essential amino acids, these are amino acids that are needed in larger quantity by the body for protein synthesis to take place. Those essential amino acids include histidine, isoleucine, leucine, methionine, threonine, phenylalanine, and valine. When you talk about non-essential amino acids, they refers to those amino acids that are needed in the body in a small quantity for protein synthesis to take place. They include alanine, aspartic acid, glutamic acid, and serine. And lastly, we have the conditional amino acids. As the name implies, these amino acids that are needed based on conditions. They include arginine, glutamine, proline, lysine, tyrosine, and cysteine. As a chain of amino acid is put together, it folds upon itself to create a three-dimensional structure. It is the shape of this folded structure that determines the functions of which it performs in the body. Information in the DNA, information is coded within the DNA by the sequence in which these bases are arranged. When you are talking about these bases, we are talking about adenine, guanine, cytosine, and thymine. That is the 4 bases of a DNA. The process of turning this coded information into protein involves 2 processes, which are transcription and translation. What is a transcription? Transcription is a process in which information coded in the DNA is transferred to the RNA, that is ribonucleic acid. RNA is a long chain of bases, just like the strands of DNA, except that it has the base uracil in place of the base thymine. There's this complementary strand of RNA called messenger RNA. What it does is that it detaches itself from the DNA, it leaves the nucleus and travels into the cell cytoplasm. After leaving the cell cytoplasm, it then attaches itself into another component of a cell known as the ribosomes. That's the site where protein synthesis takes place. And this will then lead us into another process known as a translation. Translation is a process by which the cell makes protein using the genetic information carried in by the messenger RNA. The amino acids are brought to the ribosome by a more smaller type of RNA called the transferrin RNA. So with this, I believe with all conviction that you all have understood the process of a protein synthesis. Please, if you have any question about how protein is being synthesized, you can put it down so that by the end of this class, you can be able to ask your question. Then we can now move to the next stage of our lesson, which is the main topic for discussion for the day, gene linkage. So before we say anything about it, I want us to have a look at this diagram of chromosome. These are the structure of chromosome, 3 different structure of chromosome. We have, this one is chromosome 1, this one is 2 chromosomes, but the genes are located at the separate. We have the third diagram that the first gene 1 is, the distance between gene 1 and gene 2 is very high. Then let's go back to the previous slide. 2 genes are said to be linked when they are located on the same chromosome. We assume that different chromosomes segregate independently during meiosis. What is meiosis? Meiosis is a type of cell division that involves production of 4 daughter cells, each with half number of the original chromosome of the parent cells. Linkage usually involves genes that are located close to each other. So when you take a look at this diagram, you can see in this surface diagram of the chromosome, we can see that gene 1 is very close to gene 2. The distance between gene 1 and gene 2 is very tiny. So in this case now, we can see that linkage will take place in this because the strength of linkage depends on the distance between the linked gene. So when you look at the second and the third diagram, you know that the distance between gene 1 in this chromosome and the gene 2 is very far. That is the strength between gene 1 and gene 2 is very weak. In this case now, you can say that linkage cannot occur in this second and the third diagram. There are 3 bases in which linkage can occur in a cell. We have on the basis of a complete and incomplete linkage, that is on the basis of crossing over. We have on the basis of chromosome and on the basis of genes that are involved. We are talking about on the basis of a crossing over, we have a complete and incomplete linkage. On the basis of chromosome, we have a autosomal and allosomal linkage. While on the basis of genes that are involved, we have coupling phase and repulsion phase. This is a typical example of a complete linkage. When genes that are present on the same chromosome have no tendency of separating out during crossing over, we can say that it has gone through the process of a complete linkage. We should take note, looking at this diagram, You can see the parent cells. You can see the parent cells fusing together to produce the offspring, which is the first generation. So in this case now, no recombinants are formed, only the parental combination are always observed in this. And the effect of this complete linkage is that it causes a red, green color blindness and also hemophilia. They are very common in Drosophila. We have incomplete linkage. Let's take a look at this diagram. We are talking about incomplete linkage. When genes that are present on the same chromosome have the same tendency to separate out during crossing over, we can say that it has passed through the process of incomplete linkage. And in this case now, you can see that recombinants are always formed, and it's always common in pairs and males. You can take a look at this diagram. This is the parent cell. This is a colorless shrunken seeds. This one is the gametes. So when they met, they produce this colored fruit seed. So that is the F1 generation. So for recombinants to be produced, we have to cross this F1 hybrid with this colorless shrunken seeds. And when you cross it, the process called the meiosis, which involves the production of 4 daughter cells, each with half number of the original chromosome of the parent cells. Meiosis will then take place here, and these 4 daughter cells will be formed. Then you can now cross it again. When you cross it, you are going to get your second generation. I don't know if any of you have a question. You can go to the comment section or your exercise patients so that as soon as the class finishes, I will give you a platform where you can ask your question. We have on the basis of chromosomes. On the basis of chromosome, we have autosomal linkage and allosomal linkage. When you talk about autosomal linkage, this is a type of linkage that occurs when the genes is located at the autosomes. We have allosomal linkage. Allosomal linkage is the type of linkage that occurs when the genes are located at the sex chromosomes. As I said earlier before now, there are 2 types of sex chromosome. We have the S chromosome and the Y chromosomes. We have on the basis of genes that are involved, we have coupling phase and the repulsion phase. When the dominant alleles and the recessive alleles are present on the same gene, it will show a coupling phase. You can take a look at this diagram. This one is a dominant alleles. This one is a recessive alleles. They are all lies on the same gene. So in this case now, you can see it's showing a coupling phase. You have repulsion phase. This is when the dominant allele of a gene located on the same chromosome is linked with a recessive allele of another gene on the same chromosome. In this case now, it's going to show repulsion phase. You can take a look at this second diagram. You can see dominant allele, recessive allele in the first one. Second one, you have recessive allele and the dominant allele. So in this case now, you can say that it has passed through repulsion phase. What are the significance of linkage? The significance of linkage are very enormous, but in this our class, we are going to pick few effects or significance. The first one of it is that it reduces variability, that the rate at which gametes are being formed are reduced to a drastical level. It helps in keeping the parental, racial, and specific trait together. It is also useful for maintaining the good character of newly developed variety. Linkage plays an important role in determining the nature and scope of hybridization. I believe that you guys truly understood what linkage is all about. So since there's no question, let's then move to the last part of our today's lecture, which is Mendel's inheritance. Gregor Mendel is known as the father of modern genetics. In 1856 to 1963, in 1856 to 1863, he conducted a practical on pear plants. In his search, he studied the pattern of inheritance, and then after it, he then proposed the law of inheritance, which are grouped into law of dominance, law of segregation, and law of independent assortment. In Mendel's experiment, he cross-pollinated two pure lines from constructing characteristics to obtain F1 generation. Self-pollinated F1 generation that gave rise to the F2 generation. And the reason why he chose to use pear plants, that is a Pisum sativum, to carry out this research is because it is very easy to grow and maintain, and it's an annual plant. It is naturally self-pollinated, but it can also be cross-pollinated. So these are the three laws in which Gregor Mendel proposed about inheritance. We have the law of dominance, the law of segregation, and also the law of independent assortment. In the law of dominance, in heterozygous condition, the dominant alleles get expressed. F1 generation expresses dominant alleles. We have the law of independent assortments. Two alleles do not mix when they come together in hybrid pairs and are independent of each other. And is this a law of segregation? Traits are always passed from one generation to the other. We have the law of independent assortments, which is talking about during a gamete conformation, two genes segregate independently of each other, as well as that of the other traits. So because of our time, we are going to end our lecture here. So this is room for questions. If you have any question, you can bring it up so that we can tackle it together. In our next class, we are going to talk about mutation and also a genetic frequency. So these are where we are going to draw our curtain for the day.